All right, guys, welcome back to Underground Science. And in this video, we'll be talking about the complement system. Okay, specifically, we'll be talking about the classical pathway. Okay, because there's going to be three main pathways that we'll talk about the complement system. Okay, so this video is going to be about the classical pathway. So what we should know is the complement system can get started by certain antibodies. Okay, now what are those antibodies? So we can have, let's go to the left here. We can have IgM antibodies, right? Or IgG antibodies that can start the complement pathway, or the classical complement pathway. All right, so we should know that IgM is actually a pentamer. Okay, so it's a pentamer, not like IgG. So you can actually have, it's basically five joined together, right? So it's a pentamer. This is one IgM. All right, so obviously IgM would be the most efficient activator, right? Because it's a pentamer and has multiple binding sites, okay, compared to IgG. All right, so that's basically what we need to know is that this pathway gets started by antibodies. All right, so let's start talking about the classical pathway. Let's say we have a microbe here, all right, some sort of pathogen, some sort of microbe that has antigens on its cell surface, okay? So we're going to get an antibody that's going to bind. Okay, so for example, let's talk about, whatever it can bind to any part or any antibody, whether that be IgG or IgM. Okay, so let's say we have an antibody here that will bind to the antigen. Okay, so that's the first step. So we get binding of antibody to antigen, binding of antibody to antigen. Okay, so once that happens, right, we should know next for the next step that remember the FC part of the antibody we're going to denote as being right here. Okay, that's important because the next thing that's going to happen is then we're going to get introduction of complement proteins, the first complement protein being C1. Okay, but C1 it has three different parts to it. We have C1q, C1r, and C1s. Okay, C1q will actually be the one to bind to the FC portion of the IgG or IgM, whichever antibody we're talking about here, right? So let's draw that. So if we have C1, let's just draw C1 Q like this, right? It's going to bind to the FC portion of the antibody, whether that be IgG or IgM, okay? Then the C1q portion will activate the C1r portion, right, of the C1 complement protein, which we'll draw like this. Actually, let's do a different color than orange. So let's say this is the C1r part of the C1 protein, C1 complement protein, right? So it's going to activate C1r now. Now C1r will activate, right, C1s. And that's the main part of C1 that we need to focus on. So if we drew C1s, let's say, like right here, right? We'll denote it like that. So now C1s is activated. Okay, so that was the main goal. C1q will activate C1r, then C1r will activate C1s. Why is C1s the most important? Because C1s is what's going to act as the protease. That's going to start cleaving the next two complement proteins. Okay, so now that we have C1s, right, activated, then we're going to have C4 that's going to come into play. So we said what? C1s is the protease, right? So C1s is the protease. So then we're going to have C4 that's going to be activated by C1s. So let's see how this works. So what's going to happen is C1s will cleave and activate C4, okay? Will activate C4, which will just denote it in green, or let's just do blue, C4 into two different molecules, right? So the C4 complement protein will get cleaved, right, by C1s into C4a and C4b, C4a and C4b. All right. C4b will go and bind to the pathogen surface. So if we come here, right, we're going to have C4b that's going to bind to the pathogen surface. All right, so let's just go ahead and erase this right here. C4a, right, well, just C4a is actually important for inducing mild inflammation. Okay, so we'll just write this as it'll induce inflammation. Okay, because we have to focus on C4b here. So what do we say? C4b binds to the pathogen surface. All right. And who's doing all this? C1s. Okay, it's activating and cleaving C4 into C4a and B. C4b binds the pathogen surface. Another thing that C1s will do, right, is it's going to activate and cleave C2. Okay, again, C2 will get activated and it'll cleave into two different things, one being C2a and the other being C2b. C2a will actually go and bind onto C4b, okay? So remember, we have C4b on the pathogen surface, right? So C4b is on the pathogen surface, and C2a will now come here and bind on C4b. So now we have C2a here. And then C2a, right? We said we'll bind there, right, on C4b. And then C2b will just go into circulation, right? So they'll just get released into circulation. So what we have to focus on now is this complex we just formed. Okay, this is the C4b2A complex. So what is this called? The C4b2A complex. C4b2A complex. All right, so let's make this known, okay? Because this is also going to be called our C3 convertase. Okay, so the C4b2A complex is also called C3 convertase. All right. So of course, with the name, right, we know it's going to do something to C3. So let's see what happens next. So we have C4b2A, right, on the pathogen surface, also called C3 convertase. This is a very important enzyme for the classical pathway, okay, because what's going to happen is C3 will now come by and it's going to get acted on by the C3 convertase. Okay, so we're going to have C3 floating around here, right? And it's going to get activated upon, it's going to get acted upon by C3 convertase and convert into two different things, right? Again, it's going to be converted into C3a and C3b. Okay, now C3b will go and bind onto C4b2a. Okay, so it's going to go all the way here and bind onto the C4b2a complex, which was called C3 convertase, right? So let's erase this arrow, right? Because we know where it's going now. Let's draw the arrow here. And then we said that it's going to do what? C3b will bind onto C4b2a. So now we got C3b here. So now this complex is called the C4b2a3b complex. Okay, so if we went here and wrote that down, we have the C4b2a3b complex. Okay, now this is no longer C3 convertase, of course, right? Now this is called C5 convertase. Okay, C5 convertase. Now, this is the next important enzyme that we have to talk about. Okay, so let's make this known. But before we do that, let's see what C3A is good for. Right, C3A is actually a good molecule for activating histamines, right? So this can help activate histamines, which we know, right, can lead to our vasodilation and swelling and so on. And it can stimulate inflammation. It's a potent stimulator of inflammation, all right? Which is good for attracting white blood cells to the area of where this pathogen is trying to infect, right? So now we have C5 convertase. What will C5 convertase do? C5 convertase, right, which is also called what? C4b2a3b, right, will activate and will cleave, right, C5, hence the name of C5 convertase. Okay, so let's go and write that. So this will take C5 and it'll cleave it to two different molecules, C5a and C5b. All right. Now, C5a is, again, a strong activator of inflammation. So this will stimulate inflammation, right? I don't know why these are here. Let's fix that. Sorry about that. All right, so this C5a will do what? It'll stimulate inflammation. It's a potent stimulator of inflammation, which again will help attract white blood cells to the area of where the pathogen is infecting. Okay, and also C5a, right, which we'll cover later on, can help attract and stimulate neutrophils too. All right. But C5b, though, right, is good because that's what's going to start something called the membrane attack complex. So what's going to happen is if we go back to the pathogen, right? If we draw C5b, for example, like this, right? C5b will then go ahead and bind to C6. All right, so that's going to be the next complement. C5b will bind to C6. All right, then C6 will bind to C7. Okay, then C7 will bind to C8, and then C8 will bind to C9. Okay, so C5b through C9, right? So all of this right here, if we just draw this in red, C5b, C6, C7, C8, and C9 will actually form a protein called the membrane attack complex. Why is this important? Because this will then plug in, right, to the membrane of the pathogen, and it's going to create an opening between the outside and inside environment, which will help water, which will lead to water being able to go into the pathogen, and then that will lead to osmotic lysis and cell death. Okay, so this can lead to cell death of the pathogen or bacteria, microbe, or whatever's infecting us. So that's actually pretty cool because this is how the classical pathway destroys pathogens. All right. So that's basically the classical pathway of the complement system, right? Just make sure. So the main idea is okay. We start with C1, right? Specifically C1q, which binds to the Fc portion of our antibody, that being IgG or IgM. Then we said C1q activates C1r, which then activates C1s, which then cleaves C4 into C4a and C4b. We said C4b binds onto the cell surface of the pathogen. C4a helps mildly induce inflammation. Then C1s also cleaves C2, right, into what? C2a and C2b. C2a binds onto C4b, right, which forms the C3 convertase, C4b2a. Then C3 convertase converts C3 into C3a and b. Then we said C3b binds onto C4b2a, forming C4b2a3b or C5 convertase. Then we said C5 convertase converts C5 into C5a and b. Then we said C5a induces inflammation, attracts and activates neutrophils. And then we said C5b can bind onto C6, which then binds to C7, C8, and C9, which then leads to the formation of the membrane attack complex, right? And that leads to the osmotic lysis and destruction of the pathogen or microbe that's infecting us. All right, so that's basically the classical pathway of the complement system. All right, I hope this made a little bit of sense. I hope it wasn't too confusing. Thanks for watching and see you guys in my future videos.